the role of the microbiome in health and disease, and on how the fungal component of the microbiome shapes immune responses. As you might know, the past 20 years have witnessed an explosion of publications in the field of metagenomics. And metagenomics has been so of an impact on human society that publications of metagenomics have been cited on journals like The Economist and not only Nature. But while metagenomics goes back to a statement from Hippocrates and says all diseases begin in the gut, the vast majority of what we know on the microbiome and the main results on the metagenomics evolution are related to bacteria. I would say that from 2005, so in the past 15 years, the term microbiome is present in 86,336 publications, while the term mycobiome, that is specific to the fungal component of the microbiome, is in 868, with a ratio of 1 to 100. It's interesting to notice that the ratio 1 to 100 is exactly the ratio that fungi and bacteria have in most of the environments related to the human body. So fungi and bacteria do not live alone. They are found together in a wide variety of environments, from the human oral cavity to the human lungs, to the human bowel, vagina. And they come to us, they enter our body through different ways. One of them is simply the environment. Actually, probably fungi represent much more of an environmental signature than bacteria, but others are fermented foods. And among fermented foods where bacteria and fungi coexist, I'd like to remember kefir, and wine. Why not wine or beer? There are fundamental sources of like even bread or other fermented beverages. There are sources of these microbes that enter our body via food. The approaches to the investigation of microbiome diversity do benefit enormously from the developments of the next generation sequencing technologies. And the general approach that is being used to study the fungal communities, and I discussed today, it's an approach that sees a sample preparation and DNA isolation from the sources. This is one of the critical steps, while often the methods applied to extract DNA for microbiome studies differ from the ones from a microbiome study, because fungi have a cell wall that is different from the one from bacteria, and it's usually harder to break than the bacterial one. Then, since fungi are underrepresented into the communities, both environmental and bodies, with respect to bacteria, usually it's necessary a step of DNA amplification. Why? Because if you just sequence everything using an Illumina sequencer, doing an untargeted metagenomic, most of the times you find very rarely fungal sequences. So the vast majority of the approaches as of today, unless they pass through a step of differential separation, filtration or whatever of the fungi, they actually perform DNA amplification. And the regions that are amplified in second and third generation HTS differ in size and differ in type of primers that are being used for the sequencing. The sequences undergo quality control, demultiplexing, removal of flanking genes and chimeras, and immediately we have two major problems, technical, that I'd like to mention to the public. One is the fact that often the same fungal species has marker regions. These are the standard ITS, ITS12, ITS1, 5.8S, that could differ in length in different species, and the difference in length causes often problems in analyzing the data from sequencing. And then the same strain could harbor different copies of ITS12 and ITS1 and ITS2 and the spacers that could differ both in length and sometimes in sequence. This is because often microorganisms and fungi are the results of interspecific hybridization, and there is much more of interspecific hybrids in the fungal genus of what people usually expect. This leads to the other limit that we should overcome at the moment for the microbiome studies, that is the reference database. Why reference databases for bacterial 16S or for bacterial sequences and bacterial genomes are densely populated, the fungal world represents only a small subset in the known domain. So only a very limited amount of fungal sequences, whole genome sequences, are present within the database with respect to global diversity. And this also tells where the direction of future research should go. If we look at the intestinal microbiome, we should consider that the intestinal microbiome is probably the most important organ. And I'm talking about organ, I'm talking about both a human function and the bacterial and fungal function in determining the immune responses in the human body. And the microbiome and the virome component of the microbiome are currently what I would like to call the scientific heart of darkness, taking this term from Conrad's book. Why is this? Because the bacteriome, as I said, is 10 to the 13, 10 to the 14 cells if we average to the different parts of the gut. The fungal microbiota is between the 10 to the 12th and 10 to the 13th, so at least 10 different in magnitude. And the helminths and viruses are even less known. So we are really delving into a part of the unknown that probably will lead to several surprises. These are the most common species, genuses present in the gut, but I will not read you long lists. What I want you to notice is that while fungi make up less than 0.1% of the total gut microbial communities, fungi are 100 times larger in terms of surface, maybe 1,000 times larger than bacteria. Thus, the surface with which fungi interact with the immune system is almost equivalent to the surface that the bacteria interact with our immune system. My laboratory has contributed importantly to the study of the human microbiome, and we have been working together with a small group of international researchers on this theme since 2010. So it's 10 years that we are contributing to this field. And one of the things that I think was most interesting about our discoveries was that fungi differ. It's maybe one of the most gender-related component of the microbiome, because there is a profound differences in the microbiome of males and females in terms of mean fungal isolates, so overall fungal load, and also diversity. The number of observed OTUs in females is greater than the one in males, and it's similar to the one of the child. And the other interesting point to notice is that the fungal microbiome decreases in biodiversity with aging. So the diversity and the composition in species of the fungal component decreases with aging, while for the bacterial part, this is exactly the opposite. The diversity of the bacterial microbiome increases with age. Several studies have defined a core gut microbiota that, as I said, is simplified because in reality, there is really a lot of things that we do not know, but just looking at the subset of what we do know, there are 10 major genera found in the majority of the human gastrointestinal tract. These are Candida, several types of Candida, but in particular Candida albicans in the westernized population, Saccharomyces and the Saccharomycetes, with Saccharomyces cerevisiae as my favorite subject of study, but I'll talk more about it in the next slides. Penicillium, Aspergillus, Cryptococcus, Malassezia, in particular Malassezia restricta, Cladosporum, Galactomyces, Debaromyces, and Trichosporon. Now, as you see, some of these genera are present in the environment, while some of these genera are more heavily enriched in food. In particular, Saccharomyces cerevisiae, that for several years has been described as a microorganism that was food associated or to wineries or breweries, was basically described as a domesticated organism. In this study and in a following study from our group, we actually discovered that a lot of the fungi that we're dealing with are passengers. What do I say by passenger? The only clear colonizer, the only fungus that basically stays with you all your life are the ones that belong to the Candida genus. So Candida albicans and others stay on a mouse or on a human, irrespectively of the time where you have been exposed to them. Saccharomyces cerevisiae, if you, let's say this is an experiment, that was done on mice, but also has been repeated on volunteers. If you provide a food enriched with Saccharomyces cerevisiae, fermented food with Saccharomyces cerevisiae, the cerevisiae stays within the human gut in numbers that you can count, for 2 weeks, and then it apparently disappears. In reality, we now know that it remains. It remains probably in the appendix or in remains, but if you do not supplement it through diet or with a dietary food with prebiotics that favor its growth, Saccharomyces cerevisiae becomes, basically fades away after 2 weeks of its colonization. Fungi are not alone, as we have seen. They interact with bacteria, and the interactions between fungi and bacteria see the entire spectrum of type of interactions in health and disease. So there are positive interactions and then there are negative interactions. Important studies have described how gram-positive bacteria and Candida albicans are both enemies and brothers in arms. This is a micro review that I liked a lot, but this is a mechanism of Candida albicans inhibition by lactobacilli, and it sees both a change in the pH, production of inhibitory compounds, competition for adhesion, and indirect effects. At the same time, it has been described an interaction between Saccharomyces cerevisiae and the Escherichia coli ESEC through antagonism, competitive exclusion, and host immunomodulation. And in the next slides, I will try to address how fungi and bacteria interact one with the other by means of the human immune system. So the theme I'll try to bring up is that when fungi and bacteria are within the host, they mediate their struggle or their cooperation by using the immune system of the host itself. I don't know how many of you are aware of how the fungi modulate host immunity. These are a few synthetic slides that I usually show it to my students. And so what happens is that the fungal wall component interact with several receptors on the cell, on dendritic cells. So there is TLR2 and TLR4 receptor, there is dectin-1, and there is DC sign and CLRs. And basically these receptors converge on SIC and card9, that is the signal transduction mechanism that then goes downstream to NF-κB and induces the expression of IL-12, interferon gamma, and IL-6. And This interaction occurs with dendritic cells and with macrophages, and the signals from dendritic cells and macrophages mediate the type of fate that native T cells undergo. You have generation of Th1 cells that produce interferon gamma, Th17 that produce IL-7 and IL-22, and then some signals that are mediated by IL-10, in principle, producing greater amounts by commensal fungi like Saccharomyces cerevisiae, induce the development of T regulatory cells as they suppress the production of TNF-alpha, Th17, and the major histocompatibility complex 2. While the signals from pathogens, or I would say not real pathogens, but opportunistic commensals, when they overgrow, like Candida, take the path of the IL-6 and they lead to inflammation by production of Th1 cells or Th1-derived Th17 cells in states of sustained inflammation. This is a little bit the big picture of all this story. All these responses are crucial and they have to be coordinated. They have to be coordinated in the proper way. So the activation of a Th1 response is important to lead to fungal clearance, but it has to be balanced. And the same for Th17 responses that in some amount are important for fungal clearance. They need to be balanced by T regulatory responses, because if this balance for some reason is not working properly, these might lead to fungal infections or, on the other branch, to autoimmunity. And this is what we will talk about today. But going back to the story, going back to what I told you, that I am convinced, and I really am convinced because we are studying this aspect. When we look at the interaction between fungi and bacteria in the gut, you have to consider that fungi and bacteria use the immune system along this interaction. You should be aware of a number of important papers on a mechanism that's called trained immunity. It's a memory for innate host defenses. Trained immunity can be described as fitter, faster, better immune cells, trained cells, that ensure increased responsiveness of the innate immune system to secondary stimuli after an initial encounter with a training microorganism. Now, you should know that trained immunity is currently being studied very seriously, mainly by the group of Mycobiome that actually rediscovered trained immunity. These are two reviews that I think the students should read, because they open a window on an immune part that is not so known. And why is trained immunity important? Because let's say today, in the time of a COVID pandemic, you should know that there is data supported by scientific evidence that shows that BCG vaccination, Bacillus calmette-guerin vaccination against TBC, provides some sort of defense against the SARS-CoV-2. Actually, a few scientists hypothesize this is the reason why SARS-CoV-2 is not exploding as it would have been expected in Africa. And this is a very interesting field. So as I said, BCG, Bacillus calmette-guerin, can induce trained immunity, but fungi can also induce trained immunity. The group of Mycobiome showed how Candida can induce trained immunity, and we showed how Saccharomyces cerevisiae, coming from food or coming from human sources, can induce trained immunity. So this study sees the description. It started from the initial description of fungal strains in terms of ability to induce different types of immune responses. So a lot of what I'll talk to you about in the next slides has been obtained by taking CD14 monocytes, exposing them to IL-14 and GM-CSF, producing mature disease, and setting up a screening of different strains of Saccharomyces cerevisiae, albicans, fumigatus, hyphae, cells, and pure components of the fungal cell wall, like curdlan, zimosan, mannan, and chitin, as I said, or beta-glucans, and measuring the level of cytokine production that was obtained when the disease were matured and exposed to T cells. And so you can measure the levels of production of inflammatory interleukins and anti-inflammatory interleukins, and the ability to drive production of Th1, Th17, or T regulatory cells. So what we did in my lab, let's say this is an example of this experiment. First, we exposed the human derived dendritic cells derived from human PBMCs to different strains of Saccharomyces cerevisiae and Candida, and we measured the levels of production of TNF-alpha, IL-6, and other cytokines. And then we said, okay, what happens if we pre-treat? We choose the strains that were producing a higher level of, let's say, TNF-alpha or IL-6, and the one taking lower levels of TNF-alpha and IL-6. And we said, okay, what happens if we pre-expose human dendritic cells, the training agent, and then we expose them to an infective agent? So this is basically is how a trained immunity is tested. First, you train your cells with a training agent, and then basically you simulate in vitro an infection with LPS or Candida. And as you see, they were basically chitin. The fungal chitin was maybe the strongest inducer of the trained immunity response. And then we performed another experiment. So we asked the survival of bacteria and fungi after uptake from macrophages before and after training. And we used again as training agents RPMI, cerevisiae, glucans, mannans, and chitin. And we saw that chitin was the most effective method to reduce the cell count in terms of colony-forming units of Candida albicans, Streptococcus aureus, and Escherichia coli, seen as infective agents. So basically, the ability of the cell to deal of monocytes and macrophages to deal with infective agents was increased by pre-exposition to the training agents. And we know also the mechanism. This happened because the training enhanced microbial killing by increasing the acidification of the phagolysosome. Okay, so it was more efficiently getting rid. You might say, okay, all this evidence come from in vitro experiment. What happens in real life? So what happens in real life, we perform the same experiment in mice, in a mice model, and we use the C57BL6 mice that were intraperitoneally treated at day 7 and day 4 with 1 milligram of chitin from Saccharomyces cerevisiae, prior systemic infection of 5×10⁵ Candida albicans yeast. This experiment was made in collaboration with Luigina Romani from the University of Perugia, and it was a very important collaboration for my lab because her lab has the mouse models that we were using in this study. What you see here, this is the percent of survival of controls, untreated mice. They all died within 10 days, and the survival of a mouse that was trained with the chitin from Saccharomyces cerevisiae. 80% of the mice that were trained survived the infection. This was really striking. And since we are very sensitive to limit as much as possible the use of mice, and we are very sensitive on using environmental relevant condition in my lab, we try to export this experimental system that we had developed in vitro and in the mouse also in a model that was less of impact in terms of ethics. And in our previous work, we had discovered that Saccharomyces cerevisiae is carried by crabro wasp and by Polistes dominula. These are basically wasps that are naturally present and that carry Saccharomyces cerevisiae in particular during the time of the harvest, because and it comes from the environment and gets in the gut of the animal. And so we decided two things. This was giving to us an advantage. First of all, we were not dealing with animals that were grown in a lab. We were taking the animals from the wild, and so they had been exposed to environmental conditions in different parts of the year, and we performed exactly the same trained immunity experiment that we had performed in the mouse. In order to use as a minimum number of animals with respect to what we had done in the mouse, we used the two strains of Saccharomyces cerevisiae that had been shown the better training immunity in the mouse model, and we supplemented them to the wasps. So we took 98 foundresses in autumn winter, 85 foundresses in the spring, and 53 workers in the summer. And we treated them with YP4 and YH1, that are two candidate strains inducing trained immunity. And what we did, we fed the cerevisiae and to the animals before hibernation. We perform an experiment of hibernation, so inducing hibernation, because the question was, is Saccharomyces cerevisiae protecting the animals from an infection with Escherichia coli in a model that is well accepted for insects? So we did an hibernation experiment, and then we did an infection, a hemolymph infection, inoculating 10 to the 5 cells of Escherichia coli XL1 glue, tetracycline resistance. This was a nice method because you would have an easy screen. You will simply count glue colonies on the plates. After after 24 hours of the infection with Escherichia coli on animals that woke up, we basically did an abdomen dissection, we plated, and we counted the Escherichia coli cells. In parallel, we were asking another question. What is the effect of the trained immunity, so on the submutation of Saccharomyces cerevisiae, to the gut microbiota of the wasps? So to animals that have been pre-treated with cerevisiae and animals that have not pre-treated with cerevisiae, besides Escherichia coli, they do show changes in the gut microbiota. To this aim, we extracted DNA from the gut samples of the controlled groups and the treatment groups, and we performed 16S Illumina mice sequencing. We also performed the sequence of the ITS14 to look at the fungi. Regarding the trained immunity, we had a striking result. And the striking result was that you see here the scale of CFU. Trained immunity was occurring only in the queens, only in the foundresses. The males and the workers were not protected. They didn't show a protection by pre-treatment with the fungi. And this has a great ecological significance because in this animal model, males and workers are dispensable. What really matters is the queen survives, and males and workers are so dispensable that they burn much more energy than the queens. So the protection effect was coming at a cost. In fact, after the hibernation, the protection effect was less clear, and the cost was worthy being paid for the queens, but not for the workers and not for the males. So we had a selective protection. When we looked at the changes in the microbiota in the protected versus unprotected animals, we saw that there was a dramatic change, a significant change in the gut microbiota. In the control group, in the treated group with respect to control, there was an increase in Bacteroidetes, and there was a slight increase in Fusobacteria. The treated showed an increase in Prevotella, a decrease in Staphylococcus and in Morganella, an increase in Veillonella, etc., etc. In particular, not only the Escherichia coli XL1 blue that we had given, but in general, all the microbes of the Escherichia shigella genus, the counts of the Escherichia shigella counts were significantly reduced in the treated animals with respect to those non-treated animals. Something similar was also occurring for fungi. So the administration of Cerevisiae was favoring certain fungi and disfavoring others. And an interesting result was that the pre-treatment with Cerevisiae was significantly reducing, let's say, Mechincovia. This is the most clear result that we had. We had several other species, but I'm not going to go there in detail. Now, this was telling to us that providing Cerevisiae, I'd say could be as a probiotic or through diet, alters in a very significant, almost targeted way, the composition of specific bacterial genuses and taxa. And why was this relevant? Because while we were doing these studies, both on humans and in animals, there were a few papers that were indicating that the treatment, if you look at a mouse model of Crohn's disease and adult Crohn's disease patients, the Crohn's disease microbiome had a precise signature that was seeing an increase in Candida and a decrease in Saccharomyces cerevisiae. The fact that fungi could be involved in IBD is not surprising because Crohn's disease patients, and differently from ulcerative colitis patients, show anti-Saccharomyces cerevisiae antibodies named ASCA. And so based on this evidence, we, in collaboration with Professor Leonetti for Myers Hospital, is another one of the persons that was in the initial picture that was shown by Paolo. Basically, we looked at the gut composition in bacteria and fungi in kids with early onset of Crohn. So early onset means these are kids that show sign of disease between 10 years old and 14 years old. And because we thought that the early onset could get us near to what happens early on, so which are the initial steps. This study has been published in 2 publications that I will discuss today. And now after this one, it's an environmental microbiology study. And we went further with respect of simply counting the reads or the isolates of Saccharomyces cerevisiae, albicans, parapsilopsis, or others. This is an example of, let's say, the results from the metagenomics. Actually, we did also plating to confirm the metagenomics, and this was showing that the patients, Crohn's patients, were enriched in Saccharomyces cerevisiae isolates. In particular, the patients in remission were increased in Saccharomyces cerevisiae isolates, because what I'd like to say is that all these patients were not in active disease, and a couple of them had a flare a few days after. So I mean, it was quite interesting to see the difference in the study. So this is the design of the study. We did both culturomics and metagenomics in parallel to confirm the results. And the reason why we did culturomics is that following the initial metagenomic study, we focused on a set of Saccharomyces cerevisiae strains that we had isolated from the gut of these patients. We sequenced the entire genome of a set of these strains, but in general, we characterized them also by sequencing a subset of genes that allows you to reconstruct the phylogeny of Saccharomyces cerevisiae. And we do also the macrosatellite analysis in order to compare the strains with the existing strains. And we found that the strains isolated from the gut patients did not form a unique cluster. they preferentially clustered with set of strains that not necessarily came from food origin. So they were clustering, but they were not forming a unique cluster. Clustering was probably showing their origin. But what was interesting, you can see it here because they have this YH, it means is patient age. They were showing signs of clonal expansion within the patient. So patients from one strains were clustering together. And this was a very important sign because it was showing that there was an expansion. In the most recent paper that just came out this year, published on Journal of Translational Autoimmunity, we performed immune testing in the following way. We took the patient's isolates and we took blood from volunteers, and we used both healthy volunteers and the same kids that had provided the strains. And we basically measured from the inflammatory potential of the strains that we had extracted from the volunteers and tried to correlate the results on the immune responses with the cell wall composition and the genetics of the strain we were working with. So, I mean, I'm going to go bird's eye to the main results, but if you want to read, you're welcome to read the study. What we saw is a Spearman correlation between the cell wall component and production of different interleukins. We were seeing that IL-6 production was correlated with the high levels of mannose in the cell wall, and there was a less intense but still significant correlation with glucosamine and galactose. So different compositions of the strains were associated with different levels of immune induction. We tested in a mouse model the fact that strains that were more prone to produce, let's say, interferon gamma and IL-7A were producing more severe results in the mouse model, again in collaboration with the group from Luigina Romani. So a set of the strains of the study were further tested in animal models to show that our in vitro correlation were somehow holding in an animal experiment. And then when measuring the microbiome, we saw that when you were finding in the guts, in the patient's gut, where you were finding Saccharomyces cerevisiae, you were finding an increase in bacteroidetes and a decrease in alpha proteobacteria in particular. This is, you see, these are Crohn's disease with and without Saccharomyces cerevisiae. With the Saccharomyces cerevisiae, you have bacteroidetes, and basically the opposite occurs for proteobacteria. And here you can see at the level of Enterobacteriaceae, reduced in absence of Saccharomyces cerevisiae, and the ones increased in presence of Saccharomyces cerevisiae. When you try to melt this information, so you try to make sense of this information, you cannot see at one of these traits together. So this is a review that we are currently writing on the subject, where we have taken all the publications on Crohn's disease and with patients, with mouse models, looking at the bacteria and the fungi that go up and down. The general picture that we think can explain what's going on is that, let's say, we found that in flare, so in active disease, there is an increase of Escherichia coli and Enterobacteriaceae that are reduced by Saccharomyces cerevisiae. There is an increase of Fusobacterium. There is an increase of Ruminococcus niavus, and there is an increase of Candida albicans, of Malassezia, and there is a decrease in Saccharomyces cerevisiae. And if you take together all the information I've been showing to you, this is exactly what we were seeing in the mouse model, in the insect model, and in the human model. So to us, all this picture started really making sense. And it could derive from the fact that probably these autoimmune disorders see the presence of an alteration in the membrane permeability. Everybody knows that. And when this alteration with the membrane permeability is associated to a passage through the membrane due to chronic inflammation of bacterial and fungal species that induce inflammatory responses, basically you activate a positive feedback loop towards inflammation, and you reduce the tolerogenic responses. This activation of a positive feedback loop of inflammation is such that this rule could be broken. So normally commensal should increase the levels of IL-10, IL-25 and stop production of Th17 cells. But in Crohn's disease or other inflammatory disorders, not only you have potential pathogens that induce IL-23 and IL-17 and do the proliferation of Th17 cells, but some of the commensals could end up making the same job because maybe they have an altered composition and they have an altered induction of balance of the inflammatory balance in the human body. So to conclude today's presentation, I think that systems immunology is missing really global microbiology. So we have to see at how these interactions take place, where they take place, because we know that a lot of display occurs at the Peyer's patch. And now we know that very likely this is not happening simply at the level of the gut, but it's also reflecting on the brain. A strong interest on my group is the role of fungi on the gut-brain axis. We recently published that this was a study on autism spectrum syndrome published on microbiome. We showed that autistic kids have an imbalance in the bacterial component. Again, they have a reduction in the autism disorders of bacteroidetes and increase in firmicutes. Again, you find some of the same species that we found altered in Crohn's disease. And again, you have an increase of candida counts in the autistic spectrum syndrome. This is something that links all the elements together. What I want to tell you is that a very recent paper that came out 4 months ago is showing the same type of imbalance between fungi and bacterial species that are correlated in these correlation networks in other disorders that could see an involvement of the gut-brain axis, such as Alzheimer's disease. Thus, my concluding remarks of my lecture are that fungi and bacteria live in equilibrium. When this equilibrium is altered, probably they start some sort of war, or the interaction, that are positive interaction, are completely unbalanced and altered. If this happens and occurs together with alteration of the membrane, this could lead to sustained inflammation, Th17 driven, that could lead to a number of autoimmune disorders. And for sure, there is evidence that this occurs for Crohn, for autism, and for other disorders on the gut-brain axis. I thank you for listening to my lecture, and I hope that I've been able to send the signal that is related to the importance of studying these elements together. Thank you.